form of hyaluronic acid and bacterial form of carbohydrates can have a structural group. Again, carbohydrates can have a structural group in the structure of some components. Carbohydrates can have a foil rule. They can use as a foil. For example, as you know, carbohydrates burn in our cells and produce
ATP?
ATP, very good. And produce ATP. Rather than carbohydrates, what are the second important source of fuel in our body?
Glycogen or a fat?
Fat, yes. Glycogen is a form of carbohydrate.
Yes.
So the other important source of fuel in our body are fats or lipids. Fat or lipids. Just one important thing. Our brain can only use glucose. In the fasting situation, in fasting status, do you know what is fasting?
Yes, yes.
Yeah, yes. In fasting situation, which we have no glucose in our body, in our blood,
Yes.
brain can use another substance,
Fat.
which name is ketone bodies. Ketone bodies. These substances produced from lipids in liver. So our brain can only use two things: one, glucose; two, ketone bodies. This is important, and you should keep in your mind these two types of for our brain. As I said, carbohydrate can store as a form of energy in glycogen type or starch or inulin. Inulin is a storage form of carbohydrate in potatoes, starch in plants, and glycogen in our body, in animals. We have two types of cells which store glycogen. Tissues
Muscles.
which store glycogen in our body, in humans. Muscle and liver. In your opinion, which of these tissues has more glycogen?
Muscle.
Muscle,
Yeah.
liver. Well, regarding this cell, one cell, just one cell, because the volume of the size of liver cell is much bigger than muscle cells, so one cell, one liver cell has more glycogen in contrast to one muscle cell. But with regard to tissue, because we have more muscles rather than liver cells in our body, so muscles have more glycogen rather than liver. As I said, carbohydrates involved in a process named cell identification. I give you some examples. For example, HIV virus and T helpers. For example, SARS-CoV-2 virus and pneumocytes. For example, RBC cells. So, carbohydrates in the process named key and blood involved in a process we named it cell identification. Carbohydrates can involve in another process named adhesion. We have a type of carbohydrates in our body, in our tissues. That, for example, in synovial fluid, synovial fluid. Synovial fluid is fluid in our joints. Synovial fluid has a very important role in our joint. They can make our joint to move easily without any difficulties. Like an oil. For example, an oil that you use for doors.
Yeah.
Well, so some types of carbohydrates like hyaluronic acid used in our body and in our joint to make our action, our movement more easiest. Immunological response. Again, I give you the example for RBCs. If one person with blood group of A receive a blood from a person with blood group of B, we know that these two types of groups do not match with each other.
Yes.
So that people who receive the blood group of B are in danger for dying. So, carbohydrates has an immunological group because they produce immunological reaction in our body. And the last importance of carbohydrates is the role of them in protein aging. We have a very important carbohydrate named sialic. Please write it. Sialic acid. Sialic acid is a type of carbohydrates that located on the surface of some proteins and also some cells. These carbohydrates determine the age of this protein or cells. And if the sialic acid removed from the surface, this protein or cell will be diminished, will be distracted. Will be removed in our body. Again, we have a carbohydrate in the surface of some proteins and cells named sialic acid. If this sialic acid removed from this protein or cells, that protein or cell will be removed, will be distracted, or will be diminished from our body. So this carbohydrate determine the age of protein or cell in our body. Determine the age of the protein or cell in our body. You can see here the example that I explained for you. Let me magnify the picture. As you can see here, the type of virus attached to a cell using its carbohydrates. Again, a toxin can only attach to a cell with a unique type of carbohydrate, not others. Also, we have the same situation for bacteria and other cells, which attach to a unique cell using the carbohydrates. As I explained in the first session, introduction, carbohydrate has two main functional groups. The first one is carbonic
carbonic.
group, which are a ketone group or an aldehyde. The other alcohol group is the functional group in carbohydrate or sugar. Carbohydrate has some other name, or sugars have some other. The first name for this type of substance are carbohydrates. Carbohydrates, which means that these molecules produce from carbon and water. Carbon and it means that the amount of their carbon and their H2O are the same. However, we have some carbohydrates, we have some sugars that do not have the same amount of carbon and H2O. For example, sugar without the above formula. Deoxyribose. Deoxyribose. You can see this type of sugar in which molecules? What are the main place for deoxyribose? In our
DNA, nucleus.
DNA.
Yeah.
DNA. Deoxyribose is deoxyribose find in Focus is another example. Focus is another example. The majority of carbohydrates in our body are D form. D form. Sugars. However, sometimes we have L sugars in our body. L type.
Yes.
Fructose is an example
For L.
for L type. We have also some known sugar substances with the above formula. For example, formaldehyde has the same formula like sugars. Lactic acid has the same formula like sugars, but they are not sugars. So we have some exceptions. The other name for carbohydrate is glucosides. Glucosides is a grease name and come from glycose, which means sweet. which means sweet, or saccharide, which come from saccharon and means sugar. But again, we have some sugar without the sweet taste. For example, cellulose, starch are not sweet. And also we have some non-sugar substances that are sweet. For example, glycerol, vanillin, aspartame. Aspartame is a substance that you can find it in drink, non-alcoholic drink, like cola, has aspartame. The other suffix for sugar is OSES or oses, like glucose, fructose,
Lactose.
mannose, and so on. So, each substance that has an OSE in their name is a sugar. What about classification? We have two major methods for classification of carbohydrates. The first one is based on the number of constituent units. If the carbohydrate or sugar have only one unit, we have a monosaccharide. If the sugar has 2 to 10 units of sugar, we have an oligosaccharide. And if the carbohydrate has more than 100 units of sugar, we have a polysaccharide. The other classification of sugar is based on the component of the sugar.
It's pure.
If the substance is poorly produced from sugar, not other biomolecules, or we have another molecule in the sugar structure. The other molecules include lipid or protein. Sugar plus proteins produce glycoproteins or proteoglycans. Glycoproteins or proteoglycans. Sugar plus lipid produce glycolipid or lipopolysaccharides. Do you know what is the difference between proteoglycan and glycoprotein? It's about the amount of protein and sugar. In glycoprotein, the amount of protein is higher than sugar and vice versa. For glycolipids and lipopolysaccharides, the amount of lipid in glycolipids is higher than lipopolysaccharides. Well, the first item, monosaccharide. As you can see here, the simplest sugar in nature are monosaccharides. And the simplest monosaccharides are glyceraldehyde and dihydroxyacetone. Dihydroxyacetone and glyceraldehyde are the are the simplest sugars in nature. Simplest sugar in nature. Well, tell me about the differences between these two types of sugars. What are the main differences between these two types of sugars? Aldehyde,
Ketone.
ketone. So glyceraldehyde is a type of ketones, and this one is a
Aldehyde.
aldehyde or aldose. Another one?
Isomers.
Why we have two forms of glyceraldehyde, but just one form of dehydroxyacetone?
ايه كلش الكحول.
Because we have L and D.
So why we have L and D?
الكاين كايرال.
Very good. Because glyceraldehyde has a chiral carbon in the structure. However, dehydroxyacetone
Don't
does
have.
not have don't have such
Isn't.
carbon in the structure.
Also the functional group.
Which group? Which group? Functional group, yes. Your
talked
friend
about-
Abdurrahman said about this. Well, the classification of monosaccharides. We have two type of classification. The first one is based on the number of carbon in their structure. If we have 3 carbon in the structure, we have a trioses. If you have 4 carbon, pentose, 5 pentose, 6 heptose, and 7 heptose. The second type of classification is based on the functional group. If we have aldehyde, we have aldoses, or if we have ketone, we have ketoses. What about the aldehyde? If we have a 3 carbon sugar, we named it glyceraldehyde. For ketone, dehydroxyacetone. As you can see, we have 2 glyceraldehydes, so it's right here as glyceraldehydes. But for dehydroxyacetone, you can see that the plural form uses. For tetrose, we have erythrulose. So, for pentose, ribose, xylose, ribolose, xylulose, glucose, galactose, fructose, glucoheptose, pseudoheptulose. As you can see, if we use a UL between the name of sugar and the OSE suffix, we have the ketone form of the sugar. For example, erythrose is the aldehyde form and erythrulose is the ketone.
Doctor, we're gonna memorize all of this.
Yes, it is, we can use this rule for all of this sugar. But for some sugars, we have a unique name, like fructose. Because of the importance of such sugars, we have a unique name for this sugar in our diet.
Doctor, these names, we're gonna memorize it.
Some of them are very important. For example, glucose, galactose, fructose are very important. You have to memorize such names. Again. Well, when a sugar is inside our body, which is a watery environment, the sugar is not seen in a linear form. They bend and produce cyclic form. They bend and produce cyclic form. So, in our cell, in our body, sugars are seen in cyclic form. Cyclic form. As you can see here, let me write. What? Cyclic and linear form of a unit sugar are isomers. For example, cyclic glucose is an isomer for linear glucose. Both of them are, for example, glucose. Both of them has a formula with C6H12O6, but one of them is linear and the other one is cyclic. Each sugar depends on the number of carbon can produce several cyclic forms. For example, for glucose, we have 5 or 6 ring form. The 5 types are furanose and the 6th one are pyranose. Pyranose, furanose. 3. A question. Please don't look at the board, at the picture. In your idea, which form, furanose or pyranose, glucofuranose or glucopyranose, are is the major form of glucose in our body?
The second one, 6.
Why?
Because it's 90%. 99%.
I know, but why? Let me back you to the first session. If 2 people live in this class, in this room, are moved easiest, or if 10 people live in this class?
2.
Of course 2 people, because they have more free to move easily in it. The same reason can be explained for these shapes. Of course, Pyranus has more space rather than Furanus. Therefore, Pyranus has more stability rather than Furanus. Two ring isomers should change to linear form to transform to this other ring. Each ring isomer cannot directly transform to another form. First, it should break to the linear form, and from the linear form, the other ring isomer will be produced. You can't see any flash, any arrow between these two types of isomers. It means that we should first break the ring isomer to the linear isomer and then bring another form of the isomer as I wrote here. Well, when a ring form produced, an extra property will be added to the sugar, and that is, we have now a new chiral carbon. As you know, in the linear form, the
First one.
functional carbon is not a chiral carbon, but when such sugars bend and make a ring form, now the chiral carbon and functional carbon, functional group, now is a chiral carbon. So, when a ring isomer produce functional carbon change to a new chiral carbon. Be quiet, please. Be quiet. The new chiral carbon is produced and we name it anomer. These new isomers are anomers. What is the difference between them? If the direction of OH and the sixth carbon is the same. Both of them are above the other two.
Must be open.
We have a beta isomer. If they are located in the opposite direction, we have alpha isomers. So, alpha and beta isomers is a unique sugar are anomers. Anomers. Like we know that we have diastromers, enantiomers, and now we have another type of isomers, and that is anomers. And look at this type of isomers. We have alpha D-glucose and beta D-glucose, but they have some differences with each other, and they are not the same. Although many properties of them are the same, but physical, chemical characteristics of these molecules are not the same. So these molecules are really two molecules, not the one molecule. Although we have D-glucose, but one of them is alpha and the other is beta. SubhanAllah. What a very amazing thing. In our body, the majority amount of glucose in our body is beta D-glucose, not alpha D-glucose. And it is very important when you study pharmacology in your next classes, because just one form of a drug can have a curable property, not other forms. Cellulose and glycogen has too many similarities with each other. Cellulose and glycogen have too many similarities between each other. But if you use more than 2 kilograms per day cellulose, it has no effect on your body. Your body can't absorb any glucose of this cellulose. Because the form of glucose in cellulose is not the same form that you can use in your body. You can use just one of this form of glucose, not the other form. So our enzymes adapt to use beta D-glucose more than alpha D-glucose.
Doctor,
Just let me.
because doctor is very hot. Sorry.
Hot?
We have to study now.
We have just some example. We have some example for better understanding. You should know about this example for better understanding. This is the table that I gave you in the first session of our class, and that is called classification of isomers.
Yes.
We have two types of isomers: functional isomers, stereoisomers. Functional isomers, which have different atom connectivity, like ketone and alcohol. Stereoisomer can be divided to configurational isomers and conformational. As you know, configurational isomer divided to optic isomer like enantiomer, diastomer, anomers. and ring isomers like furan and piran. We can't produce a piran form from furan form. We should break it to the linear form and then again. So we have such a configurational isomers. Again, we have conformational isomers that we do not need to break them to produce such isomers. We have envelope isomers for furans. Envelope. Envelopes mean such shape, like a letter packet, like a letter packet. Envelope. This isomer can easily be in two forms, present in two forms. The first one is such form, and the other one is this form.
Because of the
Like a closed and open envelope. Closed and open envelope. This type of isomers do not need to break to produce each other, to change to each other. They can easily changeable. Change, so they are conformation. Again, bolt and chair. This is for furans and for piranosis. We have chair and both forms. This part of molecule can easily bend to up and down without any breakage. So both and chair form of a unique pyranose are conformational isomers. Another example, functional isomer aldehydes like glyceraldehyde, erythrose, ribose, glucose, and the ketone forms, hydroxyacetone, erythrolose, ribulose, fructose. You should completely memorize these shapes, this explanation. The ketone form of erythrose is erythrolose. The ketone form of glucose is fructose. The ketone form of ribose is ribulose. Another example, the envelope isomers can easily bend up and down. Like an envelope, this part of molecule can easily bend out and chain and boat. For glucose, we have chain and boat forms. And we should know that in our body, also, although there are some conformational isomers, but we have the form, we have one of these form more than the others, and that is chain form. So in our body, the major form of glucose is chain beta D glucose. Look at the wide variety form for glucose. We have linear glucose or cyclic glucose. We have alpha glucose or beta glucose. We have D glucose or L glucose. So we can say that we can find 32
molecules
glucose in our body, just for glucose, one molecule of glucose. We have 32 types and forms of glucose in our body. 32 forms of glucose. But just one of these forms, and that is chain beta D glucose, which is the main form in our body. Why? Because our enzymes adapt to work with this type of glucose, not the other form of group. Well, that was our last slide in this section, and